Hello and welcome back. Part 4. If you notice, the number of slides drastically reduced here. I went ahead at the very end of this PowerPoint. There was actually a second PowerPoint set up as kind of cell junctions and surface modifications. So I will cover that in a separate chunk as its own thing to keep, this material all together. So polysaccharides. Polysaccharides are long-chain polymers of sugars and sugar derivatives, so stuff that's sugar, that are not informational molecules. You can essentially say most of the time they are for, I won't say most of the time they're for energy. They're for energy as well as they do some other functions like holding stuff together. We'll get to that though. They usually consist of a single kind of repeating unit, or sometimes an alternating pattern of two kinds. Repeating units of polysaccharides. That's messed up. Repeating units of monosaccharides are polysaccharides. That was reversed. There we go. Glucose also exists as a disaccharide. Disaccharide, di meaning two. So glucose exists as a disaccharide in which two monosaccharide units are covalently linked. Common disaccharide that we need to know. I'm actually going to put these on their own little slide. 100% tested right here. So we have maltose equals glucose plus glucose. Specifically, these are going to be disaccharides. Maltose is glucose plus glucose. Lactose is glucose plus galactose. And then sucrose is glucose plus fructose. There is another one. It's not usually tested. But at least not tested in this course. In future courses, I know in biochemistry, they like to ask about it. There's another one. I won't worry about it. I can't find it right away, so I'm not going to worry about it. If it comes up in another course, make sure that you pay attention to that. But 100%, these get tested all the time. Polysaccharides as energy storage. So the most familiar energy storage polysaccharides are starch in plant cells and glycogen in animal cells. Big thing, starch. Starches are for plant cells. Glycogen is animal cells and bacteria. That difference will be a test question. And be sure to read that carefully because you're looking for the word plant cell versus animal cell or bacteria. Now the best known structural polysaccharides. So we already have two components. We have two functions, right? We have energy storage, and we also have structure. The structure is cellulose, which is found in plant cell walls. Remember, cell walls, plants have cell walls. Humans, bacteria do not really have cell walls. So we have the phospholipid bilayer. Plants usually have some type of lipid bilayer, as well as a cell wall, which helps keep their more rigid composure. And mammals cannot digest cellulose, although there is kind of an exception to this. Some animals do have microorganisms that can digest this stuff, like gorillas are really cool. Now, this is just an extra thing that I've thrown in there because I think it's really interesting. This is why gorillas are able to sustain an essentially vegetarian diet and still be massive, because their bodies are able to convert all those plants to the correct types of protein. So yeah, it's pretty cool. Structural polysaccharides continuing. The best known. This is a repeat slide. Looks like. Let me. There we go. Cellulose is also known as fiber. Later on, I'll talk about soluble versus insoluble and kind of the role that they take on, but don't worry too much about that right now. And that's all you really need to know about polysaccharides. Biggest thing is this stuff right here. Not only is this tested here, this is tested in so many courses. So learn this now. Burn that into your brains. Okay, nucleic acids, DNA, RNA type stuff. So nucleic acids are the largest molecules in the body. Nucleic acid monomers are called nucleotides. They have a nitrogen base, a pentose sugar, and a phosphate group. Let me find a good example. Here we go. This guy's good. There we have our nitrogen base. So nitrogen is the base. Pentose sugar. 1, 2, 3, 4, 5. These are pentose sugars. And a phosphate group. This actually you can't really see super well. Let's do here. Let's do this image actually. Here we go. You got the sugar, the base, and the phosphate group. That's what a nucleotide looks like. The nucleic acids are of paramount importance. They're super important. Because it's our DNA to the cells, because they store, transmit, and express genetic information. It's our DNA/RNA. The two major classes are the DNA, the deoxyribonucleic acid, and the ribonucleic acid. So, a lot of information really, really quickly, okay? So, there are two types of nucleotides. We split them between purines and pyrimidines. Okay? Let me clean all of this up, make sure it looks nice and pretty. Oh, I know what happened here. I'm going to do this. Here we go. This works. Make it all look pretty. Okay, so we have our purines. Both are found in DNA and RNA. The pyrimidines we have, okay, so purines are our adenine and our guanine. Both of those are found in the DNA. So that's the deoxyribonucleic acid, right? pyrimidines, they have an extra one. So while we have cytosine and thymine, we also have uracil. Cytosine is DNA and RNA. Thymine is DNA only. Uracil, RNA only. And that replaces the thymine in RNA. Back. Again, I'll keep messing with this to make sure it looks nice and pretty for everybody. Okay. Okay. Let's do 18. There we go. Okay. We have the nucleotides. We have the nitrogenous bases. They are purines or pyrimidines. Adenine and guanine are in both DNA and RNA. Cytosine and thymine are both in DNA. Cytosine is also in RNA. Uracil replaces the thymine in RNA. We also have a pentose sugar ribose or a deoxyribose, depending on whether we're talking about RNA or DNA. We have at least one or more phosphate groups on the nucleic acid, and they pair together in this specific way. The way to remember this, the mnemonic, is at the golf course. So A binds with T, G binds with C. So adenine always binds to the thymine, and the cytosine always binds with the guanine. I'll switch these golf course. A binds to T with 2 hydrogen bonds, and G binds to C using 3 hydrogen bonds. Now also remember, if we are talking about RNA, the uracil, the U replaces thymine, the T. So that's a dead giveaway. If you're trying to figure out, is this DNA, is this RNA, look to see, is there uracil or not? If there is uracil, it's RNA. If there's not uracil, it's going to be DNA. Let me see if I want to get into... Yes, I'm going to dip my toes into this really quickly. So, with nucleic acids, when we talk about base pairing, right, that ATGC, we can figure out exactly how many of each base pair there are if we know the amount paired with it. So for example, let's say we have a strand of DNA and we say 23% of the DNA strand is G. Right? We can immediately know we can pair that with 23% of the DNA strand, because 23% is G. Another 23% has to be C. And so we look and then we figure that out. So 100% minus 23% minus 23% is going to give us 100 minus 20, that's 60, minus another 6 is 56, equals 56% remaining. So if we divide that by 2, again, because we have to have equal parts of A and equal parts of T, so 56% divided by 2 equals 25 equals 28%. So we know 28% of the DNA is A, 28% of the DNA is T. If that doesn't make sense, rewind, rewatch it. It's just understanding. You will get questions phrased sort of like this, where let's say you have this amount of percentage is this. What percentage of the DNA is whatever. You can also get questions that are asking about talking about the sequence itself. This is thinking a little bit further ahead. This was going to connect some ideas with what we call transcription and translation, and more base pairing. If I were to ask you the question, what is the matching sequence for the DNA strand? Let's say ACC TTG CTG A. Something like that. What we do is we can come down here and we can look at it. Okay, A pairs with what? A pairs with T. C goes with G. C goes with G. T goes with A. T goes with A. G goes with C. C goes with G. T goes with A. A goes with T. This is the corresponding strand that would be connected to the question strand. So see, we look and we compare the two. Later on in the future, you will get questions similar to this. You will also get questions comparing it, from going from DNA to RNA. So replacing all of the T's with U's instead. Again, this is just giving a quick introduction to this. You'll see this in the future. Okay, don't worry too much about it right now. Wait until we get deeper into the course, and you will see this. So nucleic acid uses in the cell. Big thing. This is our DNA. This is where we store our genetic information, the DNA. It's also where we can get transient expression of genetic information. What that means is because our DNA, the storage of genetic information, the DNA, is all in the nucleus of the cell, right? So how do we affect and impact, the rest of the cell? If, the rest of the cell needs to know this information. And that is through creating RNA. We make RNA from DNA in order to take that information out of the nucleus in order to get it to where it needs to go to make proteins and make other things in order to keep the cell functioning the way that it's supposed to. And we will continue to get deeper into that and what all of that means and how it all works in the future, both in this course and in biochemistry. There's a lot of overlap between these two courses. Monomeric nucleotides. Monomeric, that was that word that we learned a while back. Monomeric, let's go backwards and look and see. Remember that monomeric, meaning that it's a complete in its tertiary form. Remember what the tertiary form means. Tertiary form is referring to it doesn't combine with other strands. So it is a single-stranded polymer, right? Single strand. It's sources of chemical energy in the cell, relies usually on ATP, and cell signaling involves various nucleotides. We won't talk about this a lot. That's more in biochem and physiology that we get into that. Here is just a picture of the hydrogen bonding that goes on. The actual structure of DNA. Let me make sure I don't remember their first names. James and Francis. Okay. So the double helix model, the double helix where they actually built the DNA structure, was James Watson and Francis Crick. They call this the Watson-Crick model. Absolutely need to know that. Need to know the Watson-Crick model. They found that, the DNA is a double helix structure because that is the most we'll talk more about this in biochemistry. We'll go watch my videos in biochemistry, and as you go through it, you'll see this. You have a specific direction in which the helix turns because it is more thermodynamically stable. Okay? So that's what they discovered, that our DNA structure is this specific double helix structure. There is a hierarchy of cellular components. This is finishing the DNA. So small organic molecules are synthesized from simple inorganic substances and are polymerized to form macromolecules. The macromolecules then assemble into supramolecular structures that make up organelles and other cellular structures and ultimately the cell itself. Basically, monomers go to polymers, polymers join together, polymers, aka macromolecules. And then polymers join together, make up all the bigger and bigger structures. Just like we see here, we have these small organic molecules, make macromolecules, make supramolecular structures, build up organelles, and then finally the cell. And you could put, level 6 being whatever structure those specific types of cells makes, whether that be an organ or whatever. And then take it up another level, you have an organ system, and then another level you have the whole human being. So that right there is the end of our macromolecules lecture material. We will continue off in the next part talking about some cell junctions and surface modifications. Thank you so much for watching. Have a great day and good luck studying.